Hello everyone, in this video I'd like to talk about another commonly asked question during the PSTP interview. What you thought your most important publication was? So in order to answer this question, it's better to follow the template of abstract. So first tell them the background of your work, and then what's the problem, and what's the method you used to address the problem, and what's the result, and what's the significance. And at the end of your story, you can tell them a little bit of your future work. Just some like something in your discussion part. So now I'm going to give you my answer. Okay, Dr. Cui, can you tell me something you think is very important in your research and you published? Yeah, I definitely. I can give you. I can tell you something about that. And I think one of the most important research work I published is about the air pollution and cardiovascular diseases. And in terms of air pollution, we think the most important component is called PM 2.5, which stands for the particulate matter with the diameter lower or equal to the 2.5 micrometers. So if the size of those particles bigger than the 2.5 micrometers, after inhaling those particles, most of the particles will be expelled by the by the sputum. While if the particle size is lower or equal to the 2.5 micrometers, we think those particles can enter into our bronchi, trachea and bronchi, and further deposit into our alveolar tissue and promote our tissue secrete the reactive oxygen species as well as inflammatory cytokines to cause the systematic problem after those molecules enter into our circulating system. Well, on the other hand, we know that the bone marrow-derived bone marrow derived endothelial progenitor has ability to repair the endothelial damage or endothelial dysfunction, which is also the first step of atherosclerosis. So in our study, we think that probably the the PM 2.5 can damage can inhibit the function and the number of the bone marrow stem cells as well as EPC, endothelial progenitors, in order to accelerate the atherosclerosis. So in order to do that, we use the LDL receptor knockout mice to create the hyperlipidemic animal model, and we treat this animal with a high-fat diet and expose them with PM 2.5 as the experimental group, and we use the animals with high-fat diet without the PM 2.5 treatment as a control group. We found that following PM exposure and high-fat diet, the atherosclerosis formations significantly increased and the plaque areas also increased. And also we found that we found that the number and function of the stem cell, the EPC, significantly decreased, and the mechanism is related to the increased intracellular ROS production and inflammatory cytokine release. And by using the antioxidant and anti-inflammatory medication N-acetylcysteine, all of those deleterious effects are significantly prevented. So in this study, we described that probably we can we describe the mechanism behind the PM 2.5 in accelerated the atherosclerosis formation, and we advanced a potential treatment for the PM 2.5 related atherosclerosis formation. We may use the anti-inflammatory and antioxidant antioxidant medications to treat those patients in order to prevent this process. So that's one of my work I published. So you see that I just give the interviewer a lot of details of my work, but more but more efficient with more efficiently. I just give them actually I just follow this template and give them a little bit of the background of my story, what's the problem, what method I used, and what's the result and the significance. And also I tell them a little bit of my future work as well. So that's it. I hope you will also get something from my answer, and hope you will do well in your during your interview. So good luck.